Good morning and welcome to our Neurotech Society. I'm Elif and this is Anish.
Hello.
And today we are very excited to be speaking to Dr. Andreas Schaefer, group leader at the Francis Crick Institute, co-founder of Paradromics, and professor of neuroscience in UCL.
Hi, welcome. Yeah, thank you very much for having me here. Thank you for joining us. This is our second episode. We are going to focus more on a wider range of topics than the last episode. Those who listen will know we focused very heavily on the research side of Tim Constantino's lab. But Professor Schaefer, you have a much more diverse background than some would expect, maybe. I'm not sure if you are able to start maybe just by elaborating on how studying physics led to neuroscience and led to neurotech.
Sure, and Andreas is perfectly fine. Yeah. No, so I was always fascinated by trying to understand the brain, essentially, but it was not so clear as a high school student, as a kid, what to study. In biology, I always thought like there was a lot of memorizing plants and animals and medicine. I didn't want to be a doctor, and there seemed to be a lot of that, but I always liked physics a lot. So, and at that time, so that was, I started going to university in 1995. So in Germany, you had a lot of freedom when you studied physics. Essentially, you didn't even have to go to any classes. I mean, there were no written exams, so you just had to take a little bit of homework that you could or could not do, and then after some time, you had to sit down and do 4 sort of viva exams for your bachelor-like exam, your diploma, your pre-diploma, and they were sort of in general physics and different topics of maths. And then a couple of years later, you had to do also a couple of courses. You could choose whatever you wanted pretty much. And then you had to do your diploma, again, exams and the thesis. So you had a lot of freedom. I had done a lot of maths as a kid in school, so I didn't attend many of the maths lectures. I did other stuff I was interested in, did a lot of sort of cell biology, some other biology, some more advanced maths, advanced physics, and really enjoyed the freedom. But then when it came to deciding what do you want to research on, I actually spent my second year, I sort of was like an intern in a cell biology lab. So I learned a bit of molecular biology, cloned, learned all these sort of the techniques to get a better.
So the transition from physics to biology started in undergrad?
Yeah, I like to say that I've never made a transition to biology. I wouldn't consider myself a biologist. Yeah. I'm a neuroscientist. So my lab, I have now about 12, 13 people or so. Their background is, yes, there's biology, neuroscience degree, there's a chemist, there's a neurosurgeon, there's electrical engineering, there's biomedical engineering, there's a physicist, there's a mathematician, another mathematician. So it's very broad. Mm-hmm. So I think neuroscience, one of the many fascinating things about neuroscience is just gets so much input from so many different subjects. And I think studying physics is actually not a bad starting point, because a lot of the techniques we use, we develop, we need to apply and combine, obviously have a strong physics component. And actually in the end, understanding how networks of millions of neurons or hundreds of millions, how they interact, is essentially a applied mathematical problem. So I think the toolkit you get in studying physics, the kind of concepts, are very, very useful and very helpful. So it's never something I always was a biology interested physicist, and I did a bit of molecular work to just get to that. And then for my diploma thesis, I wanted to do a physics diploma thesis that was neuroscience. So I actually did a computational neuroscience. So I simulated how cells integrate information. And then I thought, okay, I want to really do neuroscience, but I want to be able to do experiments myself, or eventually in the lab I might have. So I convinced my diploma supervisor, who also became my PhD supervisor, let me just go to the lab we collaborate with. That is an experimental psychology lab, and learn behavior. So the very other end of neuroscience. So I went to Oxford for half a year and did lots of behavioral experiments and brought that back to the lab I was in at Max Planck Institute in Heidelberg. And then established some very quantitative behavioral work. And so my PhD was a mixture of models, so simulations, electrical recordings and brain slices, a bit of imaging to photomicroscopy, although very, very little, I should say, and behavioral work. And then I did a postdoc coming to London, to UCL, where I sort of doubled down on the in vivo physiology, because I really knew at that point I really needed to get a better experimenter as well. Mm. And now my lab has sort of gone back to that sort of somewhat breadth.
Was it like you were looking for an opportunity to get into neuroscience, as in a technology that was as fresh, as capable for new research? Was that what in vivo electroscope?
Well, I mean, I think it's a good question. I think I wanted to really understand how the brain works in its native state. Of course, I'm a very driven by physics. I'm a very sort of reductionist. I want to understand how the ion channels interact, how these beautiful structures that are individual cells, how they work. And they're much more complicated than neural network models might suggest, right? A single neuron is doing so much. But the recordings in intact animals, I think, is where you can gain information of what the brain does while something happens.
This is actually leading on to a question that we wrote earlier, actually.
We saw you in a talk in Cambridge back in January, and back then as well, you described yourself as a somewhat reductionist. So we just wanted to figure out what the meaning of that would be for you.
Interesting.
Is that dinner party talk, or is that genuinely?
No, I think reductionist in a sense that I like to think I need to understand the components and how they act together to then be able to explain how the entire structure works. So I'm not satisfied with explanations that operate on this brain region does that and it sends information here. I really want to understand, well, what does that mean? If this brain region is 100,000 cells or 1 million cells, how do these cells really do together? So my level at which I want to understand is that I want to be able to trace things back somehow to how neurons integrate information, how they interact. So not the, let's say, fMRI level of description.
Yeah.
That is very useful for figuring out what brain regions do, but not how they do it.
So on the philosophy side of it, on the reductionist sense of everything is physical, material world, would that be what you describe yourself as? The physical laws describing material matter?
That most certainly, yeah. So I think there's it's a beautiful scientific mystery how
it's a beautiful maybe
it's a beautiful scientific mystery how seemingly complex things like complex thoughts also emerge from all these individual neurons coming together.
Yeah.
But I would be incredibly surprised if it can't be explained by the physics behind how neurons membrane potential changes, how chemical messengers interact with cells.
Do you think that's an implicitly held belief by most scientists out there?
I would think I would imagine it's very difficult if you are a neuroscientist who tries to understand how the brain works, if you assume that there are fundamental things that you cannot study with the tools you have.
Yeah, it would sort of break your promises.
Yeah, but I think there are lots of ways you can say, well, the way we think of how information on single cells comes together and creates something that is simplistic. We need to develop new mathematics or new physical descriptions of things. But I don't think we need to go the excuse and say there must be something else and we can't understand this. That I don't think would be helpful if you're a scientist trying to investigate how it works.
Have you heard the is maths discovered or invented question?
Yeah.
Yeah. I don't have a strong opinion on that.
Okay, I don't know. Do you have an opinion on is math discovered or invented? I don't think I come to an answer myself.
I do think that it comes down to the question of, are we just trying to make up our own little models of the world to better understand it? And your opinions regarding that. So, for example, within a neuroscience point of view, it would be, can we understand everything that was always here? Or is it just there because it is, because it's always been there? Or is there something deeper to it that has been developing all of this time?
Well, we're getting very off neurotech and very into
philosophy.
Biggest questions out there.
I'm not sure how much I can add to that. I think certainly for my professional life, I'm strongly driven by the conviction that I can understand things with the known physics. Of course, it's like you can't understand this table we're sitting at by really deriving it from the quantum chromodynamics and quantum field theory underlying how particles interact or so. But at least, let's say, for a metal table, you can start calculating it and get to quite an accurate description of some macroscopic phenomena. In a day-to-day life, of course, things like hardness or temperature are much better concepts than talking about individual atoms. And I think that transition is one that is very difficult to make for neuroscience. We do understand individual neurons very well, how they integrate information, although not perfectly. We have some sense of how they're wired up, although we only have a statistical sense, which probably is not really ultimately helpful. If we have a complete description of a network, a complete description of the parts, then we might be able to simulate something that happens, but we still would need to really create a language, a mathematical language and description around how to create the right sort of descriptors, like the equivalent of the temperature, or maybe a little bit deeper, the phonons that are reverberating in metals as well.
I guess this actually leads me back to your research then. As someone who studied a singular part of the brain, the olfactory system in mice, so intensely, how close would you say you are to having a model of understanding the inputs, the outputs, and what's going on in there?
That's a very good question. I mean, the reason we use mouse olfaction as a model is that it's not that I'm not interested in smells, but it's not that I'm, oh my God, I really want to understand smells per se. But I do think if one wants to understand how a mammalian brain works, a mouse is a good model system because you can really interfere, you can dissect, you can train animals and so on. And now for mice, they're animals that largely operate at night, so they use their sense of smell a lot. So there's a sort of an ethological, behavioral motivation why working on smells makes sense, because it allows you to use and access an entry point into the brain that is quite natural for the animal. So you can train them quite well on smells. They use that sense. The other aspect which sort of motivated me as a physicist coming into it, as a reductionist physicist maybe, is that the anatomy of the olfactory system is quite compact. So you have receptors in the nose that send their axons into the olfactory bulb, and they make connections, they make synapses onto the projection neurons of the olfactory bulb. And those projection neurons send information to a variety of different brain regions. So already the second synapse is in either hippocampus or amygdala or from cortex. And so in brain regions to do with memory, with fear, emotions, with general space perception and so on. So you could argue that that's how I argue usually, that the olfactory bulb, this intermediate station with 1 million neurons, is actually the one region that does all the pre-processing for olfaction. Whereas for other senses, you have to actually look at quite distributed parts of the brain to get a similarly compact description. So you could argue that it's a tangible goal if you want to understand how a mammalian sense works, to say, well, we have these handful of cubic millimeters of tissue, 1 million neurons. This is kind of a size with which we can start to operate. So at this point, to come back to your question, we are getting a better and better sense of what stimuli are important, what type of smells, what structure of smells, and so on. We're starting to get to technology, and I will actually talk about that aspect of technology later a little bit as well, of how the connections are organized in this structure. And it becomes realistic to say, well, in 5 years or so, we might have a whole wiring diagram of this one brain structure. And then one can test the kind of hypothesis I just had, that if you have a wiring diagram, if you understand the biophysics of cells, you can actually sort of simulate and understand maybe the entire structure. So I think we have it will take another 5 or probably 10 years or so until we can say, well, this is a 1 million neuron, quite complete, reductionist model of the olfactory bulb, but now we can start simplifying to extract what are the important features.
And would most senses fit into that one part of the brain is purely specialized? It's a very functionally specialized sort of system, or are they more complex?
I think for we think that this first processing station, the olfactory bulb, is quite odor sense specific. But as soon as you go one connection later, even regions that are called olfactory cortex are actually, people start to accumulate more and more evidence that they are also representing all kinds of other things. So they start integrating smells with, let's say, auditory or visual or spatial inputs or so. I mean, the same idea holds true for other senses, let's say the visual system, where of course a lot of processing happens in the retina, in the eye, and then in various thalamic regions, LGN, in V1, V2, etc. But it's a bit more distributed, those specialized areas. So they're studied individual and together, but I think it's going to be more of a challenge to get a complete picture of how those senses operate.
Very interesting.
You had talked earlier about just wanting to understand what each of the individual neurons do, right?
Yeah.
So isn't that exactly what Paradromics, the company that you co-founded back in 2015, is doing right now? So I was reading up on it, and they're using direct data interface to pretty much analyze the action potentials of individual neurons. So what we really wanted to know about was just your history with Paradromics, from being a co-founder to how you're involved with it in the present day.
Right. Yeah. No. This is an exciting story for someone who really started off as as very discovery ivory tower researcher like myself. So I did my PhD in a lab that had thrived on always developing new technologies. So for my PhD advice, I got a Nobel Prize for figuring out where the patch clamp technique to record from ion channels. And when I was in the lab in sort of late 90s, people had always developed a new microscope to be able to record something they couldn't before, or to optimize some manipulators and so on. So it was very natural for me to think about building your own devices to record activity, or to build your own setups to measure behavior or measure neural activity. So when I started my lab, or actually while I was still a postdoc, I spent a summer on a fellowship in a lab of a colleague of mine in Pittsburgh, Nathan Urban, and just that we both had met while he was a postdoc, I was a PhD student, and then kept in touch when he started his own group at Carnegie Mellon, so a very engineering-heavy institution. And a year before, so we both sat in a bus, I remember, and thought like, well, we should get better ways of recording from individual neurons, really make something very tiny that could poke through and doesn't destroy the cell, doesn't die. That's it. Cut a long story short, I went to Pittsburgh for a few months to play around with focused ion beams and make tiny electrodes and try to insulate them. It was good fun. It didn't work out in the end, what I was doing there, but I met an undergrad student there who was very excited and helped me, Matt Angle. And then Matt, when I started my lab 2 years later in Heidelberg, he actually got married, took his wife, came to Heidelberg and started his PhD without speaking any word of German or ever having been to Germany, I think.
Is that by virtue of meeting you?
Well, no, he was very excited about this kind of technology development. So he pitched a couple of projects to me and said, but I really want to work on what you started in Pittsburgh. So he came to Heidelberg.
Is that a common thing in scientific worlds, to discover technology and just to want to pursue it and to move to another country, another lab?
I mean, I would think most people make the decision where to do a PhD by a mixture of projects and places where they are. I think today most institutions have PhD programs, so you apply to a program rather than to individual. At that time, in Germany, and I think partially still is the case, you can apply directly to a lab, and if the lab wants to take you, then that's how the PhD works. So it's much less structured than maybe here. But it's reasonably common that people make that decision only partially, geographically or institutionally. But for a postdoc, so the step afterwards, it's very common. I would say most postdocs can then say, well, that's my scientific career. That's the lab that I think I would want to work with, and then they figure out where is that lab, and they go. But certainly Matt took quite a leap to come to me to Heidelberg, where I was then at Max Planck Institute again. And then he tried to develop this technique to record from individual neurons, got to somewhere. It never was as scalable as we wanted, but during that process of writing up his thesis, he came up with the idea of saying, well, how could we make this approach more scalable? And then my lab actually implemented some of that while he went on to do a postdoc in a lab we collaborated with. But we stayed in touch, and at some point he decided and convinced me and Nick Melosh, his other postdoc supervisor, well, this is now, I think, an idea that lends itself to provide much more scalable brain-computer interfaces. So not record from a handful of neurons, but really record from thousands of channels ultimately, or tens of thousands.
In vivo electrode.
This is exactly so. The vision from his work in my lab was, let's develop something that's better to record from animals for animal research.
And scalability, we're talking like equivalent of the current pursuits. I don't know. I know Neuralink is doing a similar sort of thing. Other sort of in vivo electrode arrays.
So exactly. So the idea behind the technology that Matt sort of came up with during the end of his PhD, and that became the basis for Paradromics, is to essentially merge the scalability of CMOS. You know, if you think about the phone you have today compared to the phone people had 50 years ago, there's quite a significant increase in density of components, a massive scaling effect. Whereas a lot of technology, how to record from neurons in animal models, let alone in humans, has only grown very gently. And one of the reasons is that CMOS technology is just so powerful in the way it has been people have been able to miniaturize and scale. So we thought that if we can use the CMOS technology to actually record deep in the brain, if we can somehow make that work, that would be sort of leveraging all the scalability of those semiconductor silicon technologies. And the way we came up with, to cut a long story short, is the problem with silicon, with CMOS, is it's flat, right? It's very difficult to actually go into a large 3D brain with something that's flat. You can't just chuck it in and cut through it. So you need to somehow make it 3D. And so the way we came up with this is creating bundles of fine wires, so fine that they cause very little damage in themselves, and sort of press them, connect them with these CMOS chips. So you leverage the scalability of CMOS, but you make that 3D by connecting, let's say, pixels that are active amplifiers with those wires and insert them into the brain. And Paradromics has now much improved this technique, and in particular developed it more with a human application in mind. So really go for human brain computer interfaces. So all the components, all the materials used are very much those that you know that they are safe to use in human, are stable for tens of years and so on. We founded that company in 2015, I think. So started in San Jose, so in the Silicon Valley, and then moved to Austin a few years later. So now we're based in Austin, Texas, and the company has raised, I think, 18 or 20 million dollars of sort of US public money, so like DARPA, so the large DARPA grants or NIH grants, and another 86 million pounds of private money, venture capital. So now we are in the state where we have a device that is sort of in animal tests that has FDA breakthrough device designation, and the aim is to get that into patients, I think, within the next 12 months, if I'm not mistaken. But you asked about my current involvement with Paradromics, and that I should be honest, is relatively minimal. I talk to Matt very regularly, and he asks my opinion. Sometimes I might be able to help with some things, but typically at this sort of later stage of neurotech company development, it's not so much anymore about the sort of the basic neuroscience questions or the basic sort of innovative engineering question, but there's a lot about actually having solved all those. There's a lot about how do you now create devices that actually go into patients, what's the safety pathway, what's the clinical pathway, and so on, and fundraising, of course.
So obviously it's probably a sin to ask a founder where his company will go, but where do you see that technology, what do you see it being capable of when it gets into humans?
So I think the first applications of these techniques are truly patients that have severe connectivity disorders. So connectivity meaning the brain works in large parts, but it doesn't interact well with the rest of the world. And the classic examples are people that are unable to communicate, unable to talk, often late stage motor neuron disease, ALS patients. So that's the first patient group that will massively benefit from all these sort of high data rate interface. Well, I mean, all these, essentially there's a Neuralink and Paradromics. I think Paradromics probably has a slightly higher data rate. And
those are the big two.
Well, I think there are lots of other very interesting neurotech companies these days. But like companies like Synchron using a very different approach going through the vasculature. They are great. They have a very different type of invasiveness. But they also have different bandwidth they can record from. You can never record as much information from inside a blood vessel or from outside the skull or so. Or other companies that build wearables that are also quite useful for the more, let's say, consumer market. So obviously much cheaper, much less regulated, so much broader uptake. But if you want to record, I think there's now interesting in the last couple of years from sort of the basic science work of people performing recordings in patients with a variety of techniques, it's becoming clearer and clearer that increased number of neurons you're able to record from allows you to simply decode intended speech, for example, with more and more increase in word rate, so that you're not kind of decoded with 1 or 2 words per minute or tens of words per minute, but closer to the 80 or 100 words, or sometimes probably more 150 or 200 words per minute.
Sorry, you had a very good question on neurostimulation. I don't know if that was a good time to ask maybe about stimulating the brain. I don't know.
So you said that, you said earlier on that you see when it comes to actually using the product on human beings, you see that it would be used for rehabilitative purposes at first. I want to know what your opinions on using this technology for augmentative purposes would be.
It's a very good question. I think this is probably a much broader discussion. And I remember 4 years ago or so, I gave a talk at the UCL Neuroscience Symposium that was also celebrating, I think, 10 years of having a more joint neuroscience. So there was also a public outreach aspect. And I gave a public outreach talk as well on on brain computer interfaces. And I tried to make the point, at that point, no one was really talking about it very much. Like this needs more of a societal discussion. What is it that we want? I mean, what do we consider a disease or disability treated? What do we consider genuinely, desirable enhancement? I think enhancement comes with a lot of different challenges, right? It comes with the challenge of, what is it that we consider to be a human or consider us? And of course, there are lots of things how we enhance our lives that we like a lot. I mean, medication enhances life as well. The other and maybe more complex point actually is, what kind of inequalities do you start creating? True. Right? If neural enhancement is even more powerful than having access to better nutrition, then you will amplify existing inequalities that are deep across the world, but even looking at Britain today, so also very, very profound in our country here. So I don't have a clear. I think it will be inevitable that if technology is developed that helps patients to communicate the external world, that starts in the next phase starts treating patients that have maybe lost control over their limbs, which you can have a very fulfilling life without being able to walk, or even without being able to operate any of your limbs, but many have the desire to be able to become more independent, and that will those type of brain-machine interfaces will allow for that. The potentially much more impactful but also much more complex challenge comes when you're thinking not only to record and take, let's say, intended motor movement out of the brain and allow you to move a artificial arm, let's say, and to gain some independence, but when you start thinking of also stimulating the brain and possibly looking at not only these sort of more physical in inverted commas diseases, but also mental illnesses. I mean, today we, society, use brain stimulation very effectively routinely in Parkinson and different movement disorders. You can stimulate specific regions that helps dealing with tremors in an amazingly effective manner. But people are also starting to use it for severe anxiety, or severe OCD, and various other more psychiatric diseases. And I still think not sort of, it's not broadly rolled out that every hospital would do this, but there are sort of a consistently increasing number of surgeons that use that very effectively to treat those psychiatric diseases. And these are quite blunt tools in a sense. It's a single big stimulation site, or maybe a few, but a small number compared to the millions of neurons around. So if one starts having control over thousands or millions of neurons and the ability to not only record, but actually stimulate, it's potentially an extremely powerful way to deal with otherwise very, very, very debilitating mental health disorders. But of course, then you start getting into the point where, well, do I have a mental health disorder because I'm not as concentrated? Should I do that? And it's sort of similar discussions then happen around taking of specific prescription drugs for enhancing concentration. So,
well, do you see neurostimulation as potentially augmentative, as enhancing? Because obviously, reading data can be, you can control limbs that you don't even have, but could stimulation be enhancing in some way? Do you foresee a future that's possible?
I think certainly that has the potential, right? If you have the brain in different states and you want to move the brain from one state to another, I mean, one thing you can do is sleep that moves the brain from one state to another, or you could do a different meditation exercise. But if you can do some of those things, if you have network control, you could probably do them at some point also through neurostimulation, through these kind of augmentative devices. Whether one wants that or not is a very difficult question, but I think it will be very difficult to prevent that from happening if one wants to treat the sort of increasing number of mental health issues and severe mental health issues that I think are often poorly treated by pharmacological means, and sometimes impossible to treat, or only with massive side effects. And these few examples I said about deep brain stimulation, OCD, and anxiety, and various other disorders, depression. I think these somewhat anecdotal, it's a bit more than anecdotal. I don't want to pretend that this is kind of established practice, but this shows that you can control brain states in mental health disorders by stimulation. So being better and being like orders of magnitude better in stimulation, like these devices in principle would allow, although I think both Neuralink and Paradromics at this point focus on the recording aspect, but the technology platform should allow for stimulating as well.
Yeah, I suppose the issue is when you stop treating the very obviously debilitating issues and you get down to the question of, well, what is a disadvantage?
Yeah,
What
exactly.
is a problem that needs to be fixed?
Yeah. And this I do think is a debate that should rather happen earlier than later. I mean, but to put in perspective, I think the Royal Society is making great efforts. I think Tim Constantino, who you were having on the podcast last time, I think he's been involved very productively in this, I have to say. So there is a lot of sort of debate, not only around what neurotech to develop, but also, what do we want to achieve under what kind of conditions. And there are a couple of people already working on augmentation, like controlling additional arms by just using your feet or so. So it's so there are people thinking about what do patients really want. And something that I think has vastly improved in that entire space over the last years is that, unlike maybe 50 years ago, people were developing technology and then said, you patient, you need to use that. I think now at very early stages, patients are much more involved in the development, in the design of the studies. And so the question what people really perceive as a benefit to their lives comes in quite early. What I think should be a bit more at this point is not only involve patients, but involve the wider public and see like what kind of augmentations do you think are as useful as, I don't know, LASIK eye surgery, which is sort of somewhat augmentative, or simply having hearing aids or having a smartphone. What are things that we want, and what are the things where we really need to take the same decision on as many societies take on different types of drugs that some people would maybe view as augmentative, but then others look at potential risk associated with it. So, but I think this is a discussion that needs to involve the engineers, the neuroscientists, the clinicians, but it's not one that they should be only leading. I think it requires just broader.
And in terms of that jump of making a scientific discovery about stimulation into a product, is that a thing scientists do a lot? Is that I've done a PhD on a certain topic and now I want to turn it into a company?
I think that depends a lot on the culture of the institution and the lab and the group people work in. I think here at Imperial, I think there's much more of a natural thing to think about startups, to think about there's more kind of culture around pitching things to investors or having sort of small competitions and so on. Certainly when I studied physics, and when you typically study sort of more classical biology, that is only something that is an afterthought for many. But again, this is changing. The Francis Crick Institute, where I am now, we are an independent charity, but closely linked to Imperial UCL King's, as well as funded by the Medical Research Council, UKI, the Cancer Research UK, and the Wellcome Trust. But we are very discovery research institute, so people study fundamentals of how cells divide, or how parasites operate, or how the brain works. But there is a strong translational team that just talks to people, that just offers. There's a strong entrepreneurial network. Both to the public, but also to the where there are sort of close links to different VC or angel investors. But there's also simply they're there in the institute and visible and talk and encourage researchers to think about so we're ready for translation. So if there's something that becomes something that would enable a sort of funding of a startup, or would enable just translating to the clinic, there is infrastructure to do that. So I think that's a very good combination. So you don't only study things because you think this is how it then will be applied, but you say, well, this is really exciting, and then think, oh, actually, this aspect of what we're doing could have an impact in the clinic or in a business world, or actually sometimes also now in the interesting new type of institutions that sit, let's say, between a startup and a research many of these sort of focused research organizations. So just where people say, well, we come together, we get philanthropic funding and focus our work on this one big project.
Very interesting. I believe we are going to run out of time very soon. I would love to. Did you want to do this question? I think we have time to do one more question. 100%.
Do we? Okay, yeah. I'll just quickly ask. So I was reading one of the research papers that you were involved in and it was published in 2022. And basically it was a neurosurgical team study about brain-computer interfaces and both their opinions of it and their understanding. And one of the things that really surprised me about it is the people who are very much involved within this industry, they're the reason why it gets implemented in the first place. Less than 9% claims good or expert knowledge. However, reading more into it, it also showed that more than 80% of the very same people who said that they didn't have even good understanding of what was actually happening end up saying that they would trust it for rehabilitative purposes. So like, what are your opinions on the kind of disconnect between the developers of this technology and the people who apply it in a clinical setting?
So it's a very good question. I should say some other thing that has come out of my involvement with Paradromics is that group of neurosurgeons at UCL Queen Square, so it's a famous neurosurgery neurology hospital, came to me a couple of years ago and said, "Oh, we're really interested in that BCI field, and we realized that there's very little that is happening in a clinical way because there are very few brain-computer interfaces that are actually used clinically. You could argue deep brain stimulation is maybe one." And I was very excited. So we started working together a lot. Now actually one of them is sort of part-time, 50% in my lab while being 50% a neurosurgeon. And the authors on this paper, that's where that came from. And one thing we did is to start a network in London between the neurosurgeons and the neurologists and the neuroscientists and the ethicists and the engineers, because these groups don't meet as regularly. And now for the last couple of years, we've met every couple of months. I've talked originally at Queen Square, now we're moving to King's and Imperial as well. Tim Constantino is one of the sort of co-founders of this network here at Imperial as well. And we have interesting speakers from both within London as well as from all over the world that we fly in to give some talks. And the idea is exactly that, is to get the clinical practitioners to see what is the technology that is being developed, what is actually the technology we would want to be developed, and start a discussion about what the sort of the near future wins could be by bringing technology into the clinic and learning from the clinic what technology is needed. And that's very exciting. So now my lab actually has currently this one neurosurgeon, another one will join me as a PhD student, like one engineer between myself and an engineering group at King's and Östenberg. And this, I think, creates hopefully a very good ecosystem that is not only driven by one silo into another silo, but that actually things are really co-created.
Yeah.
I think, do you have a question?
I mean, what is the name of the group?
We call it Impact Neurotech Networks for implantable brain-machine interfaces for clinical translation. That's the abbreviation. So we wanted to make the point that it's not the far future brain-computer interfaces, and it's not just driven by the technology, but it's actually something for clinical use. Because my philosophy is that if one develops something that the clinicians find even a tiny bit useful, they will use it. If we can then learn from the data we get from those devices, then we can very quickly, if it's taken up very well, make next generations that get better and better.
Yeah.
Whereas if you develop something that is just better but doesn't give you more information, that will be sort of, maybe dead end is too much, but that will be useful but doesn't open up this positive feedback loop.
Yeah.
If you do something that the surgeons do not see any patient benefit, they should not really implant it typically. So so it's this combination of patient benefit and generating data that really allows us to understand more of the brain can create a very powerful positive feedback.
Yeah.
Yeah, that's perfect.
Amazing. Sorry. Yeah. Very, very interesting.
Well then, we are sadly out of time. This was
I do have follow-ups, but I have to.
Yeah.
That's it.
Yeah, I would have loved to be able to talk to you for hours on end. However, we are very much running out of time. So
But thank you for joining us on the
thank you
podcast.
so much.
Thank you for having me. That was great fun. Thank you very
Amazing.
much.
I hope anyone who listened enjoyed, and we hope they catch you next episode. Perfect.
Thank you guys.
Thank you.